Oops, that's not the right class. Here we go.
What did you get to see?
A cardio version.
Really?
Yeah.
So yeah, tell us about it.
So like there's this big guy and they like tried using this like different method of sedation for him,
Uh-huh.
and like it did not work at all.
Oh, that's
But so
terrifying.
then they did like normal sedation and like he was still like there, but like they're not forming memories. So like
Uh-huh.
you're considered sedated. And like they shocked him 3 times. And like every time he was like, oh God, that hurts so bad. But like he's not there. He just
Oh, that's stressful.
and it didn't work.
Jesus.
So like he got shocked for like
Oh,
That would, like, that's my biggest nightmare, is to be like there but not there.
you won't remember.
Yeah.
I remember I had to have an endoscopy done one time. And
that's what
I
I
was-
done and I missed the other day.
yeah. And I was talking to the doctor, you know, and I was like, well, what kind of sedation are y'all using? You know, like I'm laying on the gurney, you know, ready to go and everything. And they were like, oh, really, it's 2 different medications. And he's telling me this as he's doing it, you know, and that's all I got.
Okay.
And that was it.
When I had that 100% done, they told me, they were like, because I actually do remember, but I don't remember. Like, I
Yeah.
remember bits and pieces because it's all fuzzy.
Yeah,
Because
that's
they never
exactly
actually
right.
put me to sleep.
They never actually put you to sleep.
They twilighted me.
Oh yeah. Mm-hmm.
I was like, because apparently they took my asking questions as, oh, they want to be awake for the procedure. Because I'm just asking questions. So I have my glasses off and I'm like, what's that? Like,
Yeah.
So out of it.
Oh yeah. You're so out of it. You are so not.
And they're just like, they're answering my questions. I'm like, cool.
Yeah, I always like to ask questions. All right, so let's see. I gotta have good jokes for the end of the semester, right? So why did the cucumber blush? I told this one before. Because it saw the salad dressing.
I don't remember if you did.
Which nuts always seem to have a cold? Cashews. That's right. That's right. Let's see. Oh, I like this one. What is the easiest way to burn 2,000 calories? Leave your brownies in the oven and go take a nap. Seeing as we're talking about metabolism, that's a good one for metabolism, right? All right. So this is the culmination of everything that you have learned for 2 entire semesters. Isn't this amazing? Like, I'm so excited for this chapter. So metabolic integration, right? So just this semester, what 4 biomolecules have we talked about in terms of degradation and synthesis? Lipids, carbs, proteins, nucleotides, right? Okay. So when we talk about metabolism, which one of those 4 is not a primary metabolite for energy? Proteins. So think about, when you think about, be careful with this, right? So we got lipids, we got carbs. Proteins is really our other source, right? And then we have nucleic acids, right? So you're not really going to have nucleic acids being a primary source of energy. We talked about metabolism, how we make them, but they're not really a source of energy. And then when you think about normal conditions, what 2 sources do we normally use?
Carbs and lipids.
Carbs and lipids, right? Those are our 2 primary things. We also need protein, right? So just kind of keep in mind all the different things that we've learned, and what we're going to do now is we're going to integrate how all of them are metabolized and what signals occur in the body to shift us from one to the other, and all that kinds of stuff. So let's look at a nice big map, right? So the big 3 that we're going to talk about are proteins, carbs, and lipids, right? So when you look at what can be a metabolite for fuel. What we've looked at really is all of these pathways, but kind of in reverse, right? We're like, okay, how do we make proteins? How do we make carbs? How do we make lipids, right? That's kind of how we started each of the individual chapters. Well, now we're going to talk about basically these pathways in reverse. So what's the key metabolite in all of these pathways? Where do we kind of see a convergence of everything? Acetyl-CoA. Acetyl-CoA. So all of the primary metabolites are boxed in blue, right? So blue are primary metabolites. And so we have basically 5 big conversion pathways going on here, right? So the first one that we have is carbohydrate. So when we think about carbohydrate, what are we thinking? Carbs, I'm thinking glycolysis, pathways, gluconeogenesis. Yeah, and then glycogen is kind of a different ball of wax, but it is going to be important, right? Carbs, glycolysis, gluconeogenesis. I'm trying to make it big enough so we can see everything. All right. Our second one, let's talk about lipids. So when we talk about fatty acid metabolism, what do we call it? Beta oxidation. And then what do we call creation? We just say fatty acid synthesis. Okay, then how about the proteins? Proteins, we break our proteins down into amino acids. And so you could do amino acid degradation and synthesis. We didn't have special names for that, right? So those are our big 3. 1, 2, 3. Now, what happens when we get to acetyl-CoA? We have 2 other big pathways that happen that we kind of covered in the beginning of the year. Right. Citrate or oxidative phosphorylation. CITRate, citrate, and then we have oxidative phosphorylation. So those are our big 5, right? 1, 2, 3, 4, 5. So in all of these pathways, if we broke down proteins, carbs, and lipids, what are our primary waste products? Well, this gets turned into urea in order to be eliminated. What gets turned into urea? Why do we need urea? Nitrogen. We need to get rid of our nitrogen. What else is waste? Water's waste. Water's at the end of what pathway? Oxidative phosphorylation. What else? Think about citrate. What's a waste? What's that? What's a waste from citrate cycle? Carbon dioxide. Those are our big waste products. So all of these pathways are ways that we convert food energy, right, into ATP. That's the whole name of the game, right? So we have a lot of different ways to do that. So really we have to think about a whole organism, and we have to think about the energy balance of that organism. So you want your energy input to basically equal your energy output, right? Your expenditure. And if it's not, then you have a buildup of excess energy or not enough. What do we call the biological result of not having enough input energy? Starvation. So we're going to talk about later in this chapter, we're going to talk about kind of what happens in starvation, because we haven't really talked about that. But it's really cool. So when I took biochem as an undergrad, that was our final exam. We walked in the door and the professor had a stack of printer paper on the front desk. And he was like, everybody take a stack. And we all sat down and he wrote the word starvation on the board. And he goes, you need to write for the next 3 hours. And that was our final exam. Good and bad, good and bad. But I was like, have you seen the little meme of the little kid who's like furiously writing, you know, he's got that expression on his face? That was me. I was like, yes. Yeah. Same sort of deal. Yeah, exactly. So when we look at an entire organism, right, we have to think about all the different organs that control metabolism. So tell me, what's the primary organ? We always come back to this, like especially in our case studies, liver. We always come back to liver. Liver is so important. So all the ones that we have up on the screen here, these are our basic locations and functions for organs that play a direct role in metabolism. So this is a direct role. Right? So there can be primary, you know, organs that have a larger role than others. So some roles are major and some roles are more supplementary. So we said liver's super important, and that's very true. What else could be considered like main organs of metabolism regulation? Kidney, really important, absolutely. So we say that pancreas is supplemental. We can go with the brain. Brain's a good one. What else? So the heart's really small. It has unique requirements, right? And we'll talk about what those are because they function very differently than skeletal muscle, but you have a ton of skeletal muscle. And so your metabolism is very highly dependent upon how skeletal muscle works. So I would absolutely say that skeletal muscle is really important. So if you think, I want you to pick one more organ out of the rest of them. Just wait, sorry. It's okay. So if skeletal muscle is where you're expending a large portion of energy, where are you storing a large adipose? So that gives you the opposing sides of that coin. And the rest are kind of considered supplemental. They're still major, they're still involved, but they're kind of considered supplemental. So we'll talk about each one. So the pancreas. When we talk about the pancreas, how is it a supplemental role? Well, it doesn't actually participate in the digestion of anything, but it does produce the hormones that are required to tell cells what to do. So if you have really high blood sugar, what are you going to make? Insulin. You're going to make insulin. If you have low blood sugar, you're going to make glucagon. But that's not the only thing that the pancreas does. It also secretes proteases. And we know this already, right? We learned this not that long ago. To break down the proteins that we eat, right? You know how in our saliva we have enzymes that start to break down sugars. That's why if you get a unsalted saltine, right? So something where you're not tasting just salt, but if you take an unsalted saltine and put it on your tongue and just leave it, after time, it will start to taste sweet. Doesn't taste sweet in the beginning, but it will soon because you have enzymes in your saliva that break down the sugar. But where do we start breaking down proteins? Not until the stomach. And then you also have the pancreas that then sends additional proteases to the small intestine, right? So it's kind of like the second stop for proteins. So the ones that the small intestine makes: trypsin, chymotrypsin, elastase, and remember that those start out as zymogens, right? And have to be cleaved in order to be active. Remember that. All right, so intestines. Intestines' job is absorption, and each of them absorbs different substances. So the small intestine's job is to absorb nutrients. So have y'all done, I don't know, some people in here might have, if you've taken anatomy, have y'all done like a cat dissection? Yeah, most people have? No? Yes, okay, so some people have. So when you take out the small intestines, what's attached along the length of the small intestines? Well, the pancreas is kind of up at the top. Yes, because it secretes into the top of the intestine. But like the whole length of the small intestine, it's not just a tube with nothing attached to it. There's something that attaches along the whole side of the entire small intestine. That's the covering. That's the covering. It's really, really thin. It's really, really thin. It's like almost see-through. And if you kind of shine a light from the back, you can see it looks like branches. It's the mesentery. What's in the mesentery? No, it's connective tissue and blood vessels. Because if the intestine's job is to absorb nutrients, where do those nutrients go? You want them to go into the bloodstream. So you want lots of little blood vessels to attach to the intestines to transport those nutrients into the bloodstream. I love the cat dissection. I love it. Charles and I were walking by the door the other day when they were doing it in anatomy, and I was like, we're going in. And we did. We went in to see the cat, and he was cool with it. So, but he used to when I taught at Edie White and we do dissections on those days, if we had any left over, he'd put on gloves. He was tiny then. You know, those gloves would like dwarf him, and he would do little dissections. He's gonna be such a nerd. He's gonna be such a nerd. The other thing that the intestines will do is they will actually secrete what are called peptide hormones. Now, peptide hormones are really there to control neuronal signaling in the brain. So do you feel hungry? Do you feel full? That's what those peptide hormones help us to control our eating behaviors. And then by the time the food gets to the large intestine, there's no more nutrients, right? Food source, but we do need to absorb the water and we do need to absorb electrolytes. And how much water you absorb is very, very important, right? And if those intestines are not working, you're gonna have some serious issues. Yeah, so stomach, right? When our food, we're not going the path, we're kind of going backwards, right? We're kind of jumping around. So when we eat our food, right, goes into the stomach, and what's happening in the stomach? We know we have really low pH, right? We also know that we're going to secrete other things into the stomach, right? We know that there are cells. We're going to secrete gastrin. Gastrin is a signaling peptide. So gastrin. Anybody know any more? Secretin, R-E-T-I-N, and ghrelin, G-H-R-E-L-I, ghrelin. Those are the big three. Is it the pancreas? I'll go back and check. We'll go back and check. All right. So is it the duodenum? Duodenum or duodenum? Either one. Either one is correct. It depends on who teaches it to you, what they say. It absolutely does. Nope. You can say it either way. Absolutely. It's perfectly fine. Okay. So we know that when the food is in the stomach, right, we have very, very acidic. And what's the point of it being acidic? There are multiple. Well, so we change the pH, so we change the three-dimensional shape of proteins, right? So that we can start to denature and then eventually be subject to cleavage events, right? So if we unfold it, you're going to be able to cut it up a whole lot better. What else? Kill bacteria. What else? 3 big ones, really. Do you remember what initiates the cascade of converting zymogens to functional enzymes? A low pH, right? And then you initiate that signaling cascade that will then convert all those zymogens to active proteases. Okay. All right, liver. Kill bacteria. All right, liver. Liver is so important and so complex, right? So we call it the central processing facility, right? Our metabolic hub. Love that. But the whole point is for all of those nutrients that we just absorbed in our small intestine to basically be filtered through the liver. So the liver is going to check out the nutrients, see if they need to be detoxified, right? That's a big component of the liver that we really haven't talked about too much. And then export out what needs to be exported to different tissues, right? Because if we've been consuming a lot of sugars, right, you need to make sure that your glycogen stores in your liver are sufficient. So maybe the liver's not going to send out all the glucose. Maybe we need to replenish our glycogen stores. So the liver is going to make that decision on what is sent out to different peripheral tissues, right? The other thing is, do you remember we talked about, I think this was in Biochem 1, right? We talked about when we were talking about glucose and insulin signaling, we talked about the liver being a glucose regulator. So do you remember when we talked about how diabetics have to test their blood sugar? And we said that basically it's the same thing that occurs in the liver, right? That the liver is the one that actually determines whether insulin is going to be signaling or not based on how much sugar is out there. Do y'all remember that a little bit? That would be a good thing to go back and kind of review, right? But the whole point is for the liver to pull out excess glucose from the blood, because we don't want to have extremely high blood glucose levels. Do you want to have really low blood glucose levels? No, either way is very dangerous. And so we have to have something that can modulate that. So if it's very low, what's the liver going to do? Turn glycogen into glucose. If it's very high, what is it going to do? Turn glucose into glycogen, right? So controlled by insulin and glucagon signaling, right? But once the liver decides, okay, I'm going to export this, it's going to be exported through what's called the portal vein. So the portal vein is going to take all those nutrients to all the different parts of the body, right? So when we talk about delivery of nutrients, most things go through the liver like this, except for one. We talked about how one nutrient does not go through the liver. Do you remember which one it is? Okay, if I tell it to you, can you remind me of the pathway? How about that? Okay, it's lipids. How does it go through the body? How do we deliver lipids to peripheral tissue? We're going to go through the lymphatic system. So we're gonna import them at the small intestines, just like everything else, right? And then instead of sending it to the liver, what are we going to do with it? Carry it by albumin, right? To adipose tissue, and then adipose tissue can turn it into triacylglycerols for storage. And then when we say, okay, well, we need some fat, what are we going to do? We're going to have lipases cut it up, ship it out, right? Remember all that?
The portal vein is what they use to get the nutrients out of the liver, or
Into the liver.
From the small intestine?
From the small intestine to the portal vein, through the portal vein, excuse me, from the small intestines to the portal vein, where you're going to detoxify it, you're going to decide, should you ship it out to the peripheral tissue? Should you store it, right? What should you do with it in the liver? What is exported out of it? Multiple small blood vessels. No, portal vein goes into the liver. No. Portal vein delivers nutrients from small. So portal vein goes from small intestine to liver. Liver decides detoxify, detox, store, whatever it is that needs to be done, right? It's going to be dependent. And then you can export it out. Not through the portal vein. This is the portal vein. V-E-I-N. Come on. E-I-N. Through multiple different vessels. Okay. So let's talk about one particular molecule, right? So we talked sugar is kind of like our number one, right? So let's talk first about glucose. So if you have glucose and it's been phosphorylated, right? How did we get to that point? Well, we have, right, a dietary, D-I-E-T, dietary source of glucose. Source of glucose. Depends on where you are, right? Let's say it's delivered to the liver, right? So delivered to liver by the portal vein, vein. Then the cell is going to trap it, right? So you're going to go from glucose to glucose 6-phosphate. That's supposed to be a G, glucose 6-phosphate. So what two enzymes do that? Glucokinase. Hexakinase, glucokinase. So once that happens, we have glucose 6-phosphate, right? We're right here in the middle. How many different things could happen? 4. Looks like 4. All right, so let's go through each one. So let's say, let me zoom in. We're going to call this number 1, because this is what happens primarily. 5-glucose 6-phosphate. This is the liver. Most of it is going to go towards glycogen synthesis. So can we add glucose 6-phosphate to a growing glycogen molecule? No. What do we need to do to it? 2 things, and only one of them is on this screen. We need to turn it from glucose 6-phosphate to glucose 1-phosphate, right? So that's a mutase. And then not shown on this screen, you have to activate it by attaching it to, remember UDP? Right? Remember that? You activate glucose 1P with that. And then we can go on and we can make glycogen. Okay, so that's number 1. Number 2, let's say that there's enough glycogen made. I'm ready to release some into the bloodstream. So this is really important that some cell types have this enzyme and some don't. Do you want the liver to be able to dephosphorylate glucose? Yes, because the phosphorylation of glucose traps it in the cell. And so if one of the jobs of the liver is to get glucose out into the bloodstream, you have to have the enzyme in order to make that happen. So that's glucose 6-phosphatase, right? We know a phosphatase does what? Dephosphorylates. Okay. So where does this go? This says released into the blood. What is the primary consumer in the body? The brain. So primarily to brain. What's the other organ? It's not really an organ. The other cell type that does not want any other source of energy? Red blood cells. But brain is the primary consumer. Okay. So now let's say that we are still back, we're back in the liver, we have our glucose 6-phosphate. We've fed the brain, we've made enough glycogen, but the liver needs some reducing power, right? For biosynthesis reactions. Maybe it's making things that detoxify the alcohol that we drank. So what do we want to do? What pathway do we want to go into? What gives us reducing power? Pentose phosphate pathway. Pentose phosphate pathway. So this is 3 is if liver needs reducing power. So our glucose 6-phosphate gets converted to 6-phosphoglucanol acetone. And then we can go through the pentose phosphate pathway. When we go through the pentose phosphate pathway, we convert NADP to NADPH, and now we have our reducing power, and we can go and do biosynthetic for biosynthetic reactions. Okay, there's only one pathway left. So why would we go on this bottom pathway? There's a couple of different reasons. Be careful with feed the body, right? Feed the body is send things out, right? So we want to primarily feed the body with glucose. Well, sometimes the liver needs ATP. If the liver is going to go into biosynthesis, right? The liver needs ATP too. So that would be oxidative phosphorylation. That would be the liver needs energy. So let me zoom in on that. Okay, what else? Why else would we do this? Well, maybe we've got lots of glycogen, right? We've got enough ATP going on in the liver cell. We just have excess. So we're going to go lipid biosynthesis. This is if we have excess sugar. How about ketogenesis? When do we want to go through ketogenesis? Starvation. Right, so now we have lots of different fates. Okay, so this is kind of what happens in the liver, right? Now we have to kind of think about other tissue types. So where else do we store glycogen? Like 10% of the liver is glycogen. What's the other source of glycogen in the body? Skeletal muscle. So we're going to talk about muscle next. So we know that there are 2 types of muscle, right? Skeletal and cardiac. So I'm going to talk about cardiac kind of first, and then I'm going to talk about skeletal because we have kind of a lot to talk about in skeletal. So cardiac tissue is really interesting because think about what happens if you generally exercise. Your skeletal muscle is going to use up the store of ATP in the cell pretty quickly, right? I mean, you go run, you're going to deplete your muscle of ATP very quickly. You're going to go then to what storage form of glucose? You just told me it was glycogen, right? You just told me that skeletal muscle has glycogen. you're going to use up that, right? Now you're going to turn into a long distance runner. So you're going to have to go to yet another source. Think about the heart. Does the heart want to go through a bunch of different sources of energy? Should it ever stop? Please no, right? That's the whole point of the heart, that it always continuously beats. So if you took like a tennis ball and you squeezed a tennis ball to the same rhythm of your heartbeat, what would eventually happen? You would cramp, you would fatigue, and you would have to stop. So can the cardiac tissue have the same energy source as skeletal muscle?
No.
Absolutely not. Can't do it. Cannot do it. So cardiac muscle is going to use mostly fatty acids and ketone bodies for that reason. So you don't want it to fatigue. So it's going to use aerobic respiration as its primary source of energy. If you use anaerobic, you're going to fatigue. And that would be, that wouldn't make sense. We couldn't work, right? We just couldn't work. Skeletal muscle, on the other hand, can use a lot of different sources, right? And we learned that when we learned about our disorder, right? With the little kid who would fatigue and he would get a second wind. Why would he get a second wind? Because he would revert to another energy source, like fatty acids, like ketone bodies, right? So you have to remember that skeletal muscle is going to function in a bunch of different capacities, right? It could be that you want to sprint for a race, or it could be you want to run a marathon. Are your muscles going to work the same way if you're running a sprint than you are a marathon? No, right? And then you also have to think about, what are they going to do if you start to go hungry, right? So let's talk about skeletal muscle. Let's talk about the different kind of activities. So if we start with, for example, you're resting, right? Your muscles really aren't working too much at all. What are you going to use as an energy source? Well, you are going to use ATP. You are going to use ATP, but you're also going to use fatty acids, actually. You are. You don't need that much. You don't need a whole lot because your muscles aren't contracting a whole lot. So you're gonna use it, and then you're gonna go through citrate, right? And in citrate, we're gonna make some reducing power. What's the reducing power that we make in citrate? NADH, FAD, H2, right? For oxidative phosphorylation, okay? Now, let's say you want to run a sprint. So this is a really short burst. Sprint. Okay, so if you're talking about a couple of seconds, you want to use your ATP pool up really quickly, right? Then what are you going to go to? Your muscle cells have an extra reserve. So they have something that's called phosphocreatine. Y'all heard of this before? Okay, so we're going to talk about how phosphocreatine is actually going to contribute to ATP in the cell, right? So if you have a molecule, right, and it's phosphorylated, phosphocreatine, what we can do is take those phosphate groups and we can attach them to ADP, here's the ADP, and we can make ATP, right? So we're gonna use this as another store. So it's like a second pool of energy. When we use up the ADP, we use up the ATP, we'll use that ADP to be phosphorylated from the phosphocreatine, and your end result is you'll have ATP and you'll have creatine.
I remember that, like, if you take creatine supplements when you work out, they tell you that you have to take it every day to build up instead of like right before you work out, take it. Like it doesn't, apparently it doesn't work unless you take it like every day.
So you have to have the store already inside your cells.
So does it take a long time to like store it?
No, so I think the deal is, right, if you don't take it every day, then your body's not going to store excess phosphocreatine, right? So you have to keep giving it to your cells in order to have an excess amount of it. If you just take it and then go work out, you haven't even digested it yet. So you're not, yeah. Well, if you keep doing it on a regular basis, you can increase it, but just one time is not going to do you too much good. What's that? You got to eject it? What is that? Oh, no. Don't do that. Please don't do that. Please don't do that. Please don't do that. Okay. So if we continue running, so if we go sustained, right? We use up our ATP pool, then we're going to go to creatine, right? Okay, so now we use up our creatine pool, phospho. We're going to go to the phosphocreatine to make creatine. Then we're eventually going to switch to another energy source. Okay, so we're going to keep talking about that, right? So when you talk about using up your phosphocreatine is actually in the mitochondria. And so you actually have to have mitochondrial enzymes and cytosolic enzymes, because where do you want your ATP to be? Cytosolic or mitochondrial? Cytosolic. So there's actually a whole mechanism, and I'm not making you learn the mechanism. It's in your book, but I'm not making you know the mechanism. I'm just trying to tell you that there's an extra pool of possible ATP source. Okay. So then where do we go? We go ATP pool, we go phosphocreatine. Okay, so what's the storage glucose in muscle cells? Glycogen. Right? So what if you deplete the glycogen? You're going to go to fatty acids, just like keep going. Fatty acids, what's another one? Or ketone bodies. So fatty acids come from adipose, or ketone bodies. And ketone bodies come from where? The liver. Right? Because if you are a marathoner, right? You need to continue to have ATP available. If you stop having ATP available, what happens? You fatigue and you fall over, right? That's why have you seen that where some people, they stop running, and as soon as they stop running, they just like collapse? Yeah. All right. So muscle, think about how muscle is different from liver. Do we ever want muscle to export glucose to another part of the body? Would that make any sense for the body? No, because muscle is the primary consumer, right? So we don't want it to export it. So what you're going to see in the enzymatic makeup of the different cell types is that muscle cells can't export it. So what enzyme is going to export it in liver that we don't have in muscle? So muscle cells lack what? Glucose 6 phosphatase. Phosphatase. What else should they lack? So we don't want to export glucose. What else do we not want to waste energy making? That's true. It's not going to make ketones. That's true. What else? You don't want to make lipids. So you're gonna lack fatty acid synthase. You don't want to make this stuff. You want to use this stuff. Now, what happens if you're in starvation? What is your body going to go to for an energy source when you run out of glycogen, when you run out of fat? What's it going for? It's going for muscles. It's going for your protein in the muscles. So in extreme cases of starvation, right? So you can kind of put this on the side, starvation. Only during starvation are you going to break those proteins down to amino acids, and then the amino acids can be shipped to the liver, and the liver can actually turn it into glucose with gluconeogenesis. But that's only starvation. So you got to kind of keep that in mind, that different things are happening in the body depending on the environmental conditions. Okay.
What's that last thing you said about the liver? So the proteins are broken down into amino acids.
And shipped to the liver. Shipped to the liver. Yes. Okay. So that's skeletal. So let's talk. That's skeletal muscle, right? SK muscle. CLE. All right. So energy requirements for cardiac muscle. Do we want to keep having to switch to different things? No. So we want our heart to beat the same way for our entire lifetime. Hopefully. So we're going to use our aerobic respiration. So what are we going to do with that aerobic respiration? What do we say our two sources are? Fatty acids and ketones, comma, ketone. And from those we can make acetyl-CoA, ETY, acetyl-CoA, right? And once we have acetyl-CoA, we can go through what? Citrate and oxidative phosphorylation. So if you're going to depend on citrate cycle and oxidative phosphorylation for basically all of your energy needs, what do you have to have a lot of? Well, no, what organelle do you have to have a lot of in the cell? You have to have a ton of mitochondria. You have to have a ton of mitochondria. So mitochondria in cardiac tissue takes up like 50% of the cytosolic space. Y'all, half of the cell is taken up by mitochondria. That's cool. So, the cardiac tissue, I'll not lie, does have a really small amount of phosphocreatine, but it doesn't store glycogen. It doesn't store fatty acids. But you really want to save your glucose for the brain, right? The brain is going to be the organ that needs the most amount of glucose. You kind of think that it works the least, but it needs the most energy, but it actually does a lot of energy dependent processes that aren't movement, right? And we'll talk about that. But before we talk about brain, we're going to talk about adipose. So when we think of adipose tissue, mostly y'all are thinking about the last one, right? Fat storage. So that's true. And there are two kinds of fat storage. There's subcutaneous fat and there's visceral fat. So subcutaneous fat, where do we find subcutaneous fat? Underneath the skin, right? It's under the skin. Visceral fat, what are we talking about? Visceral. Yeah, like organ fat, like mostly abdominal. Right. But in a typical individual, it's about 15 to 25%. All the adipose tissue put together, about 15 to 20% of the person. But that's not the only thing that it does. It actually also secretes peptide hormones that regulate our homeostasis, which is super, super, super important. So when we talk about these peptide hormones, they have a huge impact on not only metabolism, but also things like immune responses also. So also immune responses, neurological development. Like there's some really interesting things. So neuro has neurological impacts, it has developmental. So things that like we don't really think about when we think about adipose. Adipose, we just think storage, right? But it has some really interesting other effects on the body. So we said two types, right? Subcutaneous. Sub-Q is just talking about, like if you're going to do a sub-Q injection, what are you doing? You're going under the skin. That's what cutaneous means. It's just skin. But just below the skin surface, right? So depending on how much subcutaneous versus visceral fat you have can actually be a predictor of cardiac, future cardiac disease. If you have more visceral, you are more likely to have cardiovascular disease versus subcutaneous. So you could have the same overall percent fat, but if more of it is visceral, then you have a higher risk of cardiovascular disease, which is kind of interesting, right? Just how your body's working. And it actually sort of makes sense just because if you have more visceral fat, it's the visceral fat that really secretes more of those hormones. And so if you alter the hormone regulation, the hormone distribution, then you got to think you're probably going to have more long-term effects on the body, right? So this is all about probably, it's still up in the air, disruption of, we call them adipokines, hormones that are secreted, peptide hormones that are secreted by adipocytes. All right, so the brain. I know you don't have this picture. I don't know what the, like this book doesn't like to put the pictures in the new textbook slides, and it drives me crazy. All right, but so the brain is where we make all of our decisions, where we control everything. So 20% of all of the oxygen that you breathe in is consumed in the brain by oxidative phosphorylation. So how active is your brain? Doesn't matter if you're sitting still or moving, your brain's working. So it's going to require a lot of glucose. It's going to require a lot of oxygen. Anything that is needed by the brain has to go through what's called the blood-brain barrier, right? I used to think, I don't know, when I would read this in the very beginning before I was actually, learned about it, I was like, okay, what is the blood-brain barrier? Like it's this thing that something has to get through, right? But the actual issue in the blood-brain barrier is you have a blood vessel. That blood vessel is carrying nutrients, oxygen, whatever it is. In order for those nutrients to get into the blood or drugs or whatever it is, they have to pass through an astrocyte. An astrocyte is a cell type, a specific cell type that attaches to not only the blood vessel, but also a neuron. And so whatever it is has to go through the astrocyte in order to get to the neuron. So there are a lot of drugs that can't pass the blood-brain barrier because they won't go through the astrocyte. It's not like delivering medication or nutrients or anything to any other cell type. It's extremely different for the brain, extremely different. And so you can see how glucose dependent the brain is if you look at a PET scan. So these are two examples of PET scans. So in PET scans, you have a glucose derivative. So this glucose derivative, instead of having an OH on one of the carbons, we're going to put a radioactive fluorine on it. And we can actually track where that goes in the brain. So you give it to the person, usually intravenously, and then you put them in a brain scan and you can see what areas light up. So the most active parts of the brain are red, yellow, right? And you get to the lighter blue, that's less active, right? So in a normal human brain, you have a large portion of the brain that's very active, right? And you can actually ask people to do things, to think about things while they're in there and light up different parts of the brain. And you can see which parts are active when they are thinking about different things. Like if you ask somebody to plan how to do something, right? You're going to have a different part of the cerebral cortex being activated, like the frontal lobe, more of it will be activated. If they're listening to something, you'll have more of the temporal lobe activated. But so it's a really interesting thing that you can do. But this picture is somebody who has Alzheimer's. This is even a mild case of Alzheimer's. What do you see different? Way less activity, right? The brain is consuming less energy overall. It's not working, right? The cells are not functioning the way that they should be. They're not consuming energy, so they're not doing their job, right? So that's a good indicator of the progression of Alzheimer's, right? You can kind of monitor that. Pretty cool, right? All right. Did I not? Okay. We talked about, yeah, we talked about that. So when you think about the brain, all of the cells not all of the cells, a large portion of the cells, right? You have neurons and then you have supporting cell types, right? And recently we've really started looking at all the supporting cell types and how influential they are in brain disorders and diseases and things like that. But if you just think about the neurons, the ones that are actually firing, right? These are going to be the ones that are consuming a ton of energy. So you have like, I don't know, 100 billion neurons inside of your brain that are firing at any given time. So how do they fire? Why do they need 60% of the body's glucose? Action potential. Why is maintaining an action potential energy consuming? What do you mean by that? So you need to have pumps, right? That you're not going to create an action potential just through simple diffusion, right? You have to actually pump against the gradient. So in order to do that, you are going to consume a ton of energy, You need ATP dependent ion pumps. And that's the sodium, potassium pump. So why can't, the brain likes glucose, right? It'll depend on ketone bodies if you're in starvation mode, but what about fats? Can the brain use fats? Not at all. Brain cannot use fats. And the reason that the brain can't use fats is these astrocytes here do not contain the enzyme required to separate fats from triacylglycerols. They can't do it. So if the astrocyte can't do it, is your neuron going to get it? Nope. So, but under starvation, like keep your kind of starvation side notes going. Under starvation, it can use ketone bodies. Good. All right, kidneys. Kidneys, you have two of them, right? And because you can survive with only one, kidneys are one of the few organs where we have live organ transplant donations and things like that, right? Most of the time it has to be somebody who is brain dead, but has enough lower brainstem activity that they're still breathing, right? And so the organs are still viable for them to be organ donors, but kidney donation can be done with a live donor. So this is one of the few. So it's the only other organ that uses gluconeogenesis. When I say only other compared to what? What's the other one? Kidney and liver. So kidney and liver. And its whole job is to pull out waste products from the blood, right? So when we talk about somebody who is in kidney failure, what do they have to go and do? They have to go and do dialysis. Well, that's what your kidneys are. Your kidneys are dialysis machines. That's what they do. So how much work does your kidney do? This is kind of an interesting thing because they're so small. People don't kind of realize how hard they work. So the average blood volume, 5 to 6 liters of blood, right, in a person, right? So how many times in one day do you think your kidneys are going to filter this 5 to 6 liters of blood? Once, twice, I think 4? 30 times per day. 30 times per day. That's a lot, right? And they're little bitty kidneys, right? They're tiny. When you did the cat dissection, everybody kind of feels like the kidneys are smaller than they should be, right? They always have a hard time finding kidneys too, because when they're shown in pictures, they kind of look like they're free floating, right? Where are they when you did the cat dissection? Who did the cat? They're like embedded on the back. Yeah, they're little, they're about golf ball size on the back in a cat, right? So they're a little bit bigger in humans, but they're like embedded, they're covered in tissue and they're anchored to the back wall, right? Or is my cat growing a tumor, right? Right, exactly. Everybody has a hard time finding the kidneys, they do. So how can we help our kidneys function? What should we be doing? Drinking lots of water. So in order to keep your kidneys working properly, you have to drink 2 or 3 liters of water a day, because you're losing volume. You're losing water volume from your blood. In what ways? Like urine, right? That's one way. Sweat. And when you exhale through the humidity, it does release water, a little bit of water vapor, not a whole lot. Not a whole lot, not much at all. But so like we were saying, that if you lose your kidney function, you can go through dialysis. Now there are 2 kinds of dialysis. The one that's shown here is hemodialysis. And in hemodialysis, you dialyze the blood. The other one is peritoneal dialysis. Anybody know what happens in that one? Anybody heard of that one? So in peritoneal dialysis, what they do is they put a port that will deliver the I always say this word wrong, dialysate. I think that's right. I think I said it right this time. They put a port on the side of your stomach. And so you put in the so see, can't say it. I'm going to say it wrong. That blood filter to remove. Yes. No, but it's, I don't know. I always say it wrong. Anyway, it's got to be isotonic to what normal blood should be. Right? So does normal blood have lots of waste product in it? No. but it is going to have things like sodium ions, potassium ions, calcium ions, 2+, magnesium ions. It's going to have glucose, right? You have to be isotonic. You're going to have your buffer system, HCO3-, right? You have to have all this different stuff that's normally supposed to be in the blood in the dialysate. So in peritoneal dialysis, what you do is you pump this same fluid into the abdominal cavity, and it basically fills the abdominal cavity. And just by diffusion, because you don't have any waste products in the solution that you pumped into the abdominal cavity, the waste products will diffuse into this now abdominal cavity that's full of fluid. And then after so many hours, you drain it. So you do it while you sleep. And you can do it at home, which is really cool.
They don't build up like fluid in their like legs and stuff from that.
It's not in there long enough for that to actually happen. Yeah. 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 But you have lots of empty space, right? That's taken up by air. And so it's not, I've heard that it's not too uncomfortable. It's not.
It's like surgery, pump air in my stomach and out.
That's super painful. That is super.
It's charged, like, not charged, not charged.
Why people, okay, if you have not had surgery in the past, when you wake up from surgery, you will be in immense pain. No one tells you that. No one ever freaking tells you that. And I remember the first time I've had 2 surgeries. I've had my gallbladder out and I've had my appendix out. And when I had my appendix out, when I woke up, I was in so much pain that I started hyperventilating because I wasn't prepared. I wasn't mentally prepared for that, right? And they were like, you have to calm down before we can give you anything. And I was like, well, this is ridiculous, you know? Right. And I think your defenses are down and you're not pre-warned. And so it's, yeah. Exactly. Because you kind of know what to expect on the second go round. So I feel like that's something that they should tell you up front. Yeah.
Because it was like an emergency surgery too, so like I didn't realize anything.
No, but I feel like just even one sentence. Now listen, right? When you come out of surgery, you're going to be in intense pain. It is going to be okay. You know, we're going to medicate you. It's going to be okay. But you do need to realize that when you wake up, you're going to be in pain. And the second time it was okay. I mean, like when I had surgery, I knew it was coming. It was fine. It was different. It was different pain because it was a lot of the air that they had put in. Did it take weeks for you?
But like it went into my shoulder.
Yeah.
Like, I'm just,
Mine was about 2 days. Oh. All right. So that was your little life lesson on the side. All right. So hemodialysis. Hemodialysis, what they do is they take blood from, usually it's from the arm, and usually there's a couple of different ways that they can do it. But they can take one vein and they kind of graft it into an artery, and they make a place where they know they're going to inject. They can insert another tube and connect it. And so sometimes you'll see patients who have hemodialysis, they have really big veins in their arms. And so a lot of those patients will like always wear long sleeves, if you've ever noticed that. So they take it out and they put it through. This is a tiny version. The dialysis machine is much bigger than that, but they pull the blood out. And so the blood is going to go through basically a tube, and constantly you're pushing fresh dialysate into the tube, right? And at the same time, the used solution is coming out. So on one side of the membrane, you have the blood that's passing through, that came out of the person's body that's passing through the tube, right? And so all of the waste products, you have a very high concentration of solutes on this side of the membrane. You have a low concentration of solutes on this side of the membrane. So things will diffuse from an area of high concentration to an area of low concentration. The things that you want to keep at a certain concentration, like sodium, like potassium, like calcium, all have to be the same on either side, right? So eventually they'll equalize. And you continue to push old used dialysate out and keep pumping fresh in and keep pumping more blood through. And then once it's clean, you can return it back to the person's body. So the slower you do this process, the easier it is on the patient. So if you can do it over 6 hours, better than doing it over 4 hours. But some people don't like to be in dialysis because you can't do this at home. You have to be at a dialysis center, right? And so you think about your restrictions on that, right? If you want to go on vacation, what's the first thing that you book? A dialysis appointment at your location, at your vacation location, right? Before you even book a hotel, you got to book that, right? Wherever you go, I got to think about, like, I would not want to travel to parts of the world that weren't as developed, because the quality of treatment is probably going to be different. So I feel like that kind of limits, what a dialysis patient can do. But well, some people, so you can't get a kidney transplant, or you've had one and you've rejected it, things like that.
But if you do dialysis and you get a kidney,
You can. Absolutely. Absolutely.
So are there people who can only can only go through it once and then they're fine, or
What do you mean only go through it once?
like go through hemodialysis, one hemodialysis treatment? And
Maybe if, let's say you, I don't know, have kidney damage, but you've not lost your kidney. In order to give your kidneys a break to recover,
right,
sometimes,
like if you've done something that's caused damage, but it's not something you do normally, it was like something that you can change.
depends on if your kidney recovers.
Yeah.
If your kidney recovers, then you can stop going to dialysis. But if your kidney doesn't recover,
Then you have to go
then
forever.
then you're going forever until you get a new one.
Or
If you
like
get a
that.
new one. Yeah. Yeah. All right. So these are all the ones that we've talked about, right? We talked liver, brain, cardiac, skeletal, adipose, and heart, right? Those are all of our top ones. So you got to think about all of these different organs have to be connected in order to maintain proper homeostasis. So we want to make sure that not only is every tissue getting the current energy needs, but all of the tissues that can store energy needs has a proper storage, right? So it's not just about immediate energy needs, it's also about storage. So of these that we've listed, there is an organ or a system that we don't list, but it is vital for maintaining metabolic homeostasis because it links all of these together. What is that? The circulatory system,
Oh, okay.
right? Yeah. Right? That's what links everything together. So you have a ton of blood vessels that have to be present in your body to connect everything. So if you put all of your blood vessels in your body end to end, right? And you got to think about, you have big ones like arteries and veins, but you also have all the way down to the size of capillaries. So if you lay them end to end, you could go around the world twice for one human. You would also die. You would also die. But that's how distributed all the blood vessels are in the body. I think it's a nice illustration, right? So what are all these affected by? Like, what are changes that can affect how these organs regulate metabolic homeostasis? How much we do, right? So like physical activity. Physical activity. What else can affect our homeostasis? So hormones are going to be secreted in response to these input things. So diet, right? So you could say like timing and extent of feeding. There's two more big ones. And we say this all the time. You guys right now are so stressed out, right? Can stress have a physiological impact on metabolism? Absolutely. So physiological stress, PHYS, physiological stress. And then somebody said genetics, right? So in a perfect world, everybody's metabolism would work perfectly, right? But is that true? No. So I'm going to say tissue dysfunction. Because maybe it's genetic, maybe it's your kidneys got damaged, right? But the end result is the same. Your tissues don't function the way they should be. So all of these are different ways that our metabolism is regulated. So this will cause changes in metabolism. Isn't this so nice to pull everything together, right?
What about like your physical environment? Not just like stress, but like, you know, hot and cold.
Sure. Sure. Yeah. Sure. It won't have a massive impact, but yeah, it will. It will alter it. So if we look kind of at all of these vital organs that we've talked about, do you notice what organ's in the middle of it all? Our liver, because that is our metabolic control center, right? So we have really 6 major functions that are required to maintain our homeostasis. So this is everything that we are doing in order to maintain homeostasis. So let's talk about liver first, right? Let's talk about liver. What are the big things that liver is going to do to take care of the rest of the tissue? Not for itself. I'm talking about for the other tissue. So what is the liver going to do for all of the other organs to maintain proper metabolic homeostasis? Export glucose. And what did we talk about in our last notes, the second to last case study? What else is it going to export? Fat to peripheral tissues, right? So here's our liver, right? We can have our glucose and we can export it out and we can send it to the brain, send it to the heart, send it wherever it needs to go. Send it to all the other tissues. And the same thing can be true about our fats. Here are our fats in red. We could send those out to the different tissue types in order to fuel their metabolism. Okay, let's do the brain next. That's number 2, the brain. So what is the brain? Now we're not talking about providing. Is the brain providing anything for metabolism other than hormones? Because that's different. No, it's not providing really an energy. It is consuming an energy. So what does it require? It requires a constant input of glucose. This is your most vital organ, right? Don't let that break. And really, it's the biggest energy drain. Okay, 3 is our heart. Again, heart's not going to provide an energy source. What is it going to consume? Really, it really requires mostly fatty acids and ketone bodies. And it can do a little, we're going to put a little glucose. A little glucose. Oh, and that's time. Okay, so what I gave y'all, a little handout, and it's asking some really big questions. Like those handouts, the questions look short, but they are really big idea questions. So we haven't gone through all of them, but what I kind of want you to do is I want you to take them home and look at them and just start jotting down some notes so that when you all get together and work on it together next time, that you have stuff to contribute, okay? It is on Moodle too. Yes, it is on Moodle. It says like chapter 19 handout.
So what do we need to study for fun?
So yes, it is cumulative, but chapter 19, when you go through and you're reading it, right? A lot of times it's really helpful to go back in the other chapters. So primarily primarily the exam is going to focus on things that are in chapter 19
That also focus on other.
that we've already covered.
So is any of
You
that?
see what I'm saying? So am I going to put photosynthesis on the final exam?
Oh, thank God.
No, I'm not going to put photosynthesis on the final exam.
Thank
So
God.
use chapter 19 to know what to study in previous chapters.
Gotcha.
That's a basis as a study.
I hate photosynthesis.
I was about to say, like,
That's terrible.
don't put it back to photosynthesis.
Okay, Maris, you need anything?
But it is helpful to go back and look at. like,
Yes,
the-
now go back in the book, go
to
back.
Look at the pentose
Okay,
phosphate
good.
pathway.
All right, I'll see you next time.
Glycogen.
Yeah, go back to Pentose